Students, this is your quick podcast about some of the genetic vocabulary that you'll use throughout this unit. We're just going to simply write this down in the word inventory rather than give you some other note sheet. So just make sure you get your definitions and your pictures as you follow the podcast. So when we look at just what genetics is, right, genetics is just the study of how genes are passed on from parent to offspring. And if we look at that term gene, we kind of throw the word heredity in there. So heredity is just defined by how different genes are passed down from parent to offspring. So we can really say that genetics is the study of heredity if we wanted to make that a really shortened sentence. Um, an example of the science of studying these inherited patterns of traits is through Mendel's work. So Gregor Mendel studied heredity in pea plants and made some really important observations. And he was just making observations about physical traits. Of course, they didn't have all the gene technology to study that, but he was able to simply make observations based on the physical characteristics, and that really led everyone else down the line. Or really started the idea of where the existence of genes, where genes came from, and that led to eventually the discovery of DNA and the double helix. So all this was vital to really studying DNA in the future. So if we back up just a little bit, chromosome review, just to remind you what we talked about last unit. Humans have 23 pairs of chromosomes, 46 total, and keep in mind that a human gets 2 copies of each chromosome. So this is actually a copied version. Remember, this is half of this is a chromosome, and when it's been copied, you have a sister chromatid. So this would be after the S phase, where the DNA has been copied. But just keep in mind that every human gets 1 chromosome from each parent, all right? And on those chromosomes, you have genes, and these are the directions for making proteins. These are smaller segments of DNA that code for proteins that eventually cause your traits, right? And again, when we say smaller segments of DNA, it's a sequence of DNA. The letter combinations, the code that actually will code for things like eye color, type of hair, seed shape, or pod color. And so Gregor Mendel came along in the 1860s and was interested in how traits specifically were inherited. So he was trained in science and math, and he understood the mathematics of probability. So he started to just cross, when we say cross, look at the pea plants and how they crossed with one another, or mated with one another, or pollinated one another, so they could produce offspring. And he just recorded in a notebook essentially what he found when he crossed certain pea plants together. Keep in mind that in genetics there's a lot of probability, and we're really looking at the number of events that are completely random. So that the chance that event A is going to happen, or the chance that event B is going to happen, it's really the number of events that meet your requirement divided by the total number of possible events. So for instance, if you tossed a coin, there's 2 options, right? So if you wanted to know what is the probability I'm going to flip heads, you have 1 for heads, 2 for the total, and that's a 50% chance. Same thing with a deck of cards. If you wanted to, what is the probability of drawing an ace, you have 52 cards in a deck, and there are 4 aces in a deck, so you just divide 4 by 52, and it's about a 7.7% chance of, on any random deck that's shuffled, that you would pick an ace out of the deck properly. So that probability leads us to genetics about the chances of getting certain traits, and that helps scientists when it comes to definitely a lot about disease and certain inherited traits they can plan for the future. So Mendel was working with pea plants, and he allowed the plants to self-pollinate for many generations, and that allowed him to produce true redeemed plants, or plants that would only produce one type of plant. And he set up 3 generations of plants. The P1 generation was the parent generation, the F1 were the offspring, and the F2 were the offspring of the offspring of the F1. So when we do Punnett squares and such, we use this terminology, P1, F1, F2. So refer back to this, or write this down on your notes sheet, just exactly what P1, F1, and F2 stand for. But again, P1 parent, F1 offspring, F2 offspring of the offspring. And over the years, when he recorded all this data, he came up with these laws, and these were pretty consistent throughout his observations, and he was able to say that it's a law because he, again, consistently collected the same type of data. So what he found was that the law of segregation, and these are, again, to remind you that alleles separate during the formation of your gametes in meiosis, and only one allele for each trait will be passed on to the offspring. So again, you are making, in meiosis, your haploid cells. So everyone is diploid, right? They have two genes or two chromosomes from each parent, and only one of those is going to be passed on to the gametes. So the fact that they separate, those alleles separate, it's called the law of segregation. The law of dominance just simply states that some alleles are dominant over the other, which means that some genes are going to be expressed more than other genes. The law of independent assortment says that allele pairs separate independently in anaphase. And we'll get back to that. But one thing that's not in your notes is this law of dominance. If you wanted to add that to your vocab sheet, that might be really helpful. So here's what he observed, and this tool right here is a Punnett square, is what we use to determine probability, but he observed these traits in the grandchildren. So he started off with two purebreds in the P1 generation, a purple flower and a white flower, and he crossed them together, and they were all purple. But when he took some of the children, the F1, and he breeded them, then he got a white plant. So, and again, the first generation, they were all purple, but that white plant didn't kind of show up until the grandchildren. So it skipped the generation. So he had some questions. What does that mean? And it starts with alleles, okay? And again, the alleles, what he found is that alleles are different versions of a gene. So if you have a gene for flower color, there was two different colors. There was the purple and there was the white, right? Some genes have more than two, but for the instance of this simple dominance, we found that there are two. So you have big F, and we represent one with a capital and one with a lowercase, which we'll get to in a minute. But again, purple flowers had the big F, white flowers had the small f. All right, and here's just another individual example of chromosomes and the different versions of the alleles. So what he found again was that there was this dominance and recessive. So which one actually won out? And the dominant allele is always expressed over the recessive. All right, it just sort of wins. So the dominant alleles are always shown with a capital letter, and in pea plants it was the purple flower. So whenever this allele was present, regardless of what the second letter was going to be, or the second allele, the dominant trait always wins. Okay. The recessive is the allele that's masked, right? It's not observed when two different alleles are present. So we represent that with a lowercase letter. In order to actually show the recessive trait, both of the alleles must be recessive, because if one of these, and again, you get one of these chromosomes from each parent. So again, this is the diploid version of an individual. So if one of these letters was the capital F would win, meaning that it would be a purple flower. But in this case, because both of the letters are recessive, then we get the white flower. There's different terminology we use to look at the combinations of genes. So for instance, when you have two letters that are the same, when both of the alleles are the same, whether it's two capital letters or two lowercase letters, we call that homozygous. All right, homozygous means pure. Sometimes you hear that as purebred, but you can either be homozygous dominant or homozygous recessive right here. Okay, homozygous dominant means that you have two capital letters. Homozygous recessive means you have two lowercase letters. Heterozygous is often referred to as a hybrid, and that's when both of the letters are different, right? Hetero meaning different. So sometimes we call these carriers as well. So you have one capital and one lowercase, but in this instance, in simple dominance, not always, but in simple dominance situations, the capital letter wins, right? So if this is tall, the big T would be tall, little t would be short, tall would win. So you'd see a tall individual, even though that individual is a carrier and could have children possibly that are short, that individual is tall. Genotype, when we refer to an individual's genotype, these are the actual letter combinations that we are referring to. Homozygous dominant, homozygous recessive, or heterozygous. Those are the letter combinations that make up the trait. Okay. When we are talking simple dominance, there are only three possible genotypes. You either have a homozygous dominant, a heterozygous or a homozygous recessive. The phenotype, on the other hand, is what you actually see. These are the words we use to describe the individual based on their traits. So I can't tell when someone is tall. I see that they're tall and they might have the dominant trait, but I don't know if they're homozygous or heterozygous. All right, so the phenotype is just simply using the words to describe the traits. What do they look like? What does the individual actually have? Right? So our important question, how do we know which genes we will inherit from our parents and which genes will show up in our bodies? If your mother has blue eyes and your father has brown eyes, how do we know what eye color that you're going to have or the baby's going to have? And the simple answer is that we use something called a Punnett square. We can use Mendel's laws of probability and our knowledge of how genes are inherited, and to use a Punnett square to really kind of make that prediction of what are all the different possibilities. I know even when it comes to blood type that I have a blood type, I'm type AB, and my wife is type O, and then we can do a Punnett square to figure out what our children were going to be. So it's very useful in figuring out probabilities of genes, and that leads a lot of doctors in the medical field a chance to figure out these genetic disorders that could potentially be passed on from parent to offspring, and not just in humans, but in plants and other animals as well. So we're going to get into some of this Punnett square stuff in the future, but all we want to know now is just the basic terminology that you need to know to be successful in this unit.